Lasergene Genomics includes all of our tools for next-gen sequence assembly and analysis. Most projects begin with our highly accurate assembler, SeqMan engine. Perform de novo assembly and reference-guided alignment for whole genomes and exomes, transcriptomes, metagenomes, and more. The software also supports data from all major next-gen sequencing technologies and can assemble multiple samples from a single project. Beyond assembling and mapping read data, the software automatically includes tasks like variant detection, CNV calculation, RNA-seq normalization, and ChIP-seq peak detection as part of the assembly process. Our downstream analysis tools allow you to perform multi-sample genome scale analysis on data for genomic variation, gene expression, and gene regulation, using multiple interactive views, clustering, and filters. Locate important genes, variants, peaks, and other regions of interest using ArrayStar's robust search tools, including our integrated variant annotation database, which allows you to locate important variants in human datasets based on population genetics, functional predictions, conservation, pathogenicity, and clinical significance. The software also includes easy-to-use methods to examine read alignment and variant calls. And with GenVision Pro, you can easily view and compare assembly coverage across the genome between multiple samples. You can also use GenVision Pro to analyze multiple project types together. For example, a combination of RNA-seq and ChIP-seq peaks. Lasergene Genomics can also be used for a variety of clinical research applications, supporting both Mendelian and cancer gene panel analysis, association studies, validation control, and Sanger validation workflows.